Welcome everybody to the last day of September meeting of the IMAG MSM Working Group on viral pandemics and multi-scale modeling. Today we have Kaiming Ye talking about the vaccine development subgroup efforts, and I hope the discussion on this topic, maybe some other discussion as well. I hope that the slightly elevated level of meeting security didn't cause people problems. If you're here, I guess by definition, that means you were able to get in. So maybe that's a pointless statement. I appreciate your being here. I need to remind you, as usual, that the meeting is recorded, it's live streamed on YouTube, and will be made available for future use. As always, Reinhardt and myself are available for comments, questions, or suggestions. Jim, Luke, and Bruce are also available to help out for anything we need. We have our usual communication channels. Somebody's actually using the Slack channel, so that's nice to know. Please help us get the word out for our meetings and others. I'm still looking for people to help me annotate the YouTube videos. So if somebody feels like spending a few hours helping annotate YouTube videos, I'd be most grateful to you. So now I will come to the announcements. Jim, I think you wanted to remind people about your survey.
Right. So if you haven't done it yet, please fill out the little survey. The URL is in the slide, which doesn't help anybody. There it is in the chat. It's only 7 or 8 questions. So if you get a chance, please fill it out. So far, we've got about 75 responses. So we're missing about 75 responses. So please fill out the survey.
I know I don't want to steal Jim's thunder, but it was quite impressive that almost half of the people said that they'd either started working on a collaborative paper or a collaborative grant based on their interactions in the working group. And so our group may not be huge, but the fact that that many people are actually doing things based on it, I think is impressive. Appreciate that. Okay, so now Reed, did you want to talk about that RFA just briefly, and then we can talk more about it next week if you want.
Sure. So there's a RFA in slide. I just put a link in the chat. As you can see, it's systems biology for infectious diseases. This is a U19, and it's got a it's a combination of projects and cores, and one of those cores is a modeling core to integrate systems data to predict the onset and severity of infectious diseases. And as I mentioned, there will be it's a long FOA, but take a moment to look through it, and there should be a Q&A posted in the next couple of days. And so perhaps after that comes, if you still have questions, feel free to reach out to me. Thanks.
Okay, thank you very much. Any other announcements? I should say that I'm still trying to hire possibly 2 postdocs in multi-scale modeling, agent-based modeling. So if you have any grad students who are graduating or postdocs who are looking for extra postdocs, I'm looking to hire 2 people in the areas we work in here. For upcoming meetings next week, we have David and Shelby O'Connor talking about 2 rather different public health questions. We have Balas Matas talking on October 14th. There's an opportunity if somebody wants to fill in that second slot, we can do that. If not, we'll have a shorter meeting. October 21st, we have Jason Shoemaker talking, and then also work on IQ cell. And we're looking for future speakers. I think, Bruce, we have a fair number of people booked in late October, early November, but there's still quite a few open slots as well. Again, please think laterally. If there's an experimentalist or a clinician you'd like to hear from, please give us the names and we'll try to bring them into the fold to speak. Any comments on the agenda going forward that people would like to make before I move on? No. Okay, this is probably a more open meeting than usual. So muting isn't that important, although I guess if you're not speaking, please mute. And I don't think we really have to hold questions since it's a discussion session today. And so I will turn the floor over to Kaiming. And thank you again, Kaiming, for being willing to speak.
Thank you. See if I can do the, okay, let me see, I can share my screen. All right. See, I can, all right, they should be fine. Okay, thank you so much for the very kind introduction and good afternoon. So this is Kaiming Ye, and I'm the professor, department chair, biomedical engineering at Binghamton University. In the next maybe 30 minutes, I'm going to introduce part of the work on the vaccine development. We're talking about the two vaccines we developed in the last 10 years. One is to basically, the vaccine was developed against the H5N1 influenza virus. And our recent works more focused on develop the vaccine for treating the cancers, basically for immunotherapies. We developed the preventive and also therapeutical vaccines for treating the cancers. So this is the first talk more focused on the yeast vaccine. So this is the vaccine was developed based on work we have done back to about 20 years ago. And that time we developed the technology to display the protein on the surface of the yeast. This is the technology, this panel summarized the principle of the technology we developed. So this is the yeast. Yeast has the wall. So we basically genetically engineer the proteins which have the anchor, which called the GPI anchors here. And this anchor will allow this protein to be mobilized on the surface of the yeast. Then the domain of the protein will expose to the extracellular space. In that way, we can functionally display the protein on the surface of the yeast. So this is basically to demonstrate the effectiveness of the technology. We simply display the GFP, that's the fluorescence, green fluorescent protein on the surface of yeast. The reason we try the GFP first, because they will allow us to calculate, to determine whether the protein surface display technology works. So this is a panel to show the surface display, the GFP. You see the green, that's the fluorescence emitted from the GFP on the surface of the yeast. And the red color indicating we use the antibody against the GFP to further confirm and that's not autofluorescence. This is basically the GFP displayed on the surface. This basically is a confocal on the microscope imaging that showing the very clearly there's the circle or we call the green, the bell of the green fluorescence, and on the surface, not basically in the intracellular. So that's basically clearly indicating this confocal microscope, clearly indicating the protein was displayed on the surface of the yeast, not intracellular space, not extracellular space. Then in order to further confirm the surface display of the GFP, we did a cell fractionalizations. We collected the different fraction of the cells. So in this case, we take the sample from the medium. We did not detect any presence of the GFP in the medium. That means the GFP was not secrete into the extracellular space. We collected the cell-free extract, and then we did not detect the presence of the GFP in the cell-free extract. That's indicating the GFP actually was transported to the cell surface, not retained inside the cell, inside the intracellular space. The GFP we can detect it is in the cell wall fractions. So that's basically fractionalizations, very clearly indicating the localization of the GFP on the surface of yeast. That's indicating the technology worked. To use this technology to develop the vaccine, we need to first successfully or functionally display the HA. That's basically hemagglutinin. Hemagglutinin. That's one of the antigen on the virus. That's the H5N1 virus. So this is the one trigger the immune response and from the host. So we want to see if we can successfully display this the hemagglutinin of the H5N1 virus on the surface of yeast. The next step, we see if this recombinant yeast can be used as a vaccine to elicit the protective antibody production. And later, we want to show whether both the humoral and also the cell-mediated immune response occurred in the vaccinated, the mouse can protect the mouse from in being infected by the H5N1 virus and during the virus challenge experiment. So after we display the HA on the surface of the yeast, we first using the fluorescence microscopy to determine the success for display of the HA on the yeast. This is the green. That's basically using the physics conjugate to the antibody to detect the surface display of the HA. You will see the HA very nicely displayed, was very nicely displayed on the surface of the yeast. Then we use the flow cytometry to quantitative analyze to follow the time course of the HA display on the yeast. So we look at the 24 hours, we don't see any expression. Then 48 hours, we see little expression. 70, this is the top panel, that's control. That's basically the yeast was transfected with the dummy plasmid does not code and the HA and genes. So the bottom panel, it shows the yeast transfect with the HA display the plasmid. So you see on the top panel in a negative control, we did not detect any HA on the surface of the yeast as expected. So bottom panel, we start to detect the display of the HA because we use the antibody against the HA, and then we use flow cytometry to analyze the antibody labeled the yeast. So if you look at a 24-hour, we detect a 41% yeast and display the HA. 48 hours, 72 hours, we see the increased number of the yeast that display the HA on their surface. So that's another indication the success for display of the HA on the surface of the yeast. So next we have to determine whether the antigenic site of the HA preserved on the yeast surface after display. So we use the two receptors. One is called an avian receptor. We know the H5N1 is avian influenza virus. We also tried another human receptor. So we want to see the surface display the HA can recognize these two receptors, can recognize both the avian receptor and also human receptors. So we did a control. If the green color shows control, so the control means that the yeast does not display the HA. Obviously, those yeast will not recognize the avian or the human receptors. We use the positive control. That's basically H5N1, inactivated H5N1 vaccine. That's basically virus. We see they recognize both the avian and also human receptors. If you look at the surface display the yeast, this line, and show this surface display the HA, can recognize both the avian and also the human receptors. So that means the antigenic site of the HA was preserved on the surface display the HA. So if you look at the degree of the binding, and certainly the HA, the surface displayed HA, more preferred or binding much stronger than the avian receptor than the human receptor That's basically expected because this is the avian influenza virus. But on the conformation of the binding to the human receptor, that's basically the huge conformation of the success of the experiment. So the next, we want to see if the surface display, the HA surface displayed yeast can trigger the response. In other words, the antibody productions. We detect the IgG and the IgG production and after vaccination. So we tried the two different immunomethodology. One is the muscle injection, the other the periodontal injection, the vaccines. So we do the primary injection, then also the boost, basically 2 shots. So the boost occurred 2 weeks after the first shot. So here show you we collected the blood sample and detect the IgG titer in the blood we collect at a different time point. So if you look at the sample, this is basically serum. That's basically its negative control. We use the dummy plasmid, also transfect the yeast is another negative control. So we couldn't detect any IgG, HA specific IgG and from those vaccinated mouse, and that's basically is obviously. So we also use a positive control. That's basically is the inactivated H5N1 virus is a vaccine to inoculate into the mouse. So we detect a high level of the IgG produced after boost, basically second shot. So then we also detected the high level of the IgG production in the HA surface displayed yeast, the vaccinated mouse. So this is basically very clearly indicating the success of the immune response, the induced after the vaccination with the HA surface displayed the yeast. So we don't see too much difference for the different route and the muscle injection and periodontal injection, the all IgG production and level, and in the vaccinated mouse doesn't show there's significant difference. So this is basically the bracket bar shows basically after 5 months. So this is basically in the day 28. This is the 5 months. So that 5 months we still can detect the quite a high level of the IgG maintaining in those vaccinated mouse. This is basically is a good result, and that's indicating not only the IgG induced right after the boost, but also can last at least 5 months. That's basically is very critical for vaccination to provide the protective vaccination and against the influenza virus. My throat freezed. Okay. So next test. So we want to see whether the IgG titer is sufficient to protect the mouse and from the virus infections. So this is the basic number. We did a HI titer. So we know that if HI titer can reach above 40, then consider sufficient to protect from influenza virus. Then we look at the titer here, and we almost detect the 128 HI titer, and in the HA surface display, the yeast vaccinated mouse against this is basically the host virus we used. We also detect the cross-reactivity. We see whether this vaccine can provide the different virus. Then we use the basically duck. And this is basically the H5 virus discovered in 2006 and 2002. We see the reduce of the HI titer in those against those virus, because in the H5N1 virus in 2004 virus, we detect 128 titer, HI titer. For 26 and 22, the titer reduced to 40, but still sufficient to provide the complete protection, because we know the 40 is the number we want to see in those fully vaccinated mouse after the second boost, after the boost. So that's basically this test. This test suggests not only the vaccine can against the host virus, but also can against the other the different variants. That's basically indicating there is the IgG induced by the HA surface displayed yeast can provide a cross protection against the different variant, because in this case we tested the H5N1 isolate from 2006, and 2002. Now we try to looking for whether the vaccine can induce another part of the immune response. That's basically cell-mediated response. So in order to confirm that response, we detected the production of the interferon gamma and also interleukin 4 production in the mouse, in the vaccine in the mouse. Now we see there is a difference. We see the very high level production of the interferon gamma and interleukin 4 in the muscle injected the mouse, and compared to the interferon gamma and also interleukin 4 production in the periodontal injected mouse. So this is the difference, and explain the reason later on. We explain to you why we did this assay. What does assay mean for the protection of the for protection from the influenza virus? So, and then we start to basically to challenge the mouse, to do the virus challenge experiment, to see actually the virus work, to see actually the vaccine works to protect the mouse from the H5N1 infections. So we use the clinical score. That's basically is to indicating how healthy of the mouse after infected by the virus. So if you look at it here, this is basically show you for the unprotected mouse, basically inoculates with the serum and also dummy and plasmid transfected yeast. And we see the score very, very high after challenge. For vaccinated mouse, and there's a clinical scores close to 0. That means those mouse did not develop any symptom of their influenzas. So, and then we're looking at another indicating, we look at a weight loss. So weight loss is another indication to see the healthy status of the mouse and after virus challenge. So we see for unprotected mouse, and then the weight after virus challenge. So they lost the weight very dramatically, and they all die on the day 6. For vaccinated mouse, and the weight basically pretty much remained very constant, and the mouse basically lasts, and for almost 14 days, we didn't see any the deaths of the mouse. Almost 100% of the mouse will protect from the virus infections. So we also did the survival rates and the determination. So we see almost 100% of mouse dies in this unprotected mouse after virus challenge, and 100% of the mouse will survive and after the vaccination, after the vaccinated. So that means there's a vaccine provided almost 100% of the protections. So the other study we did, and basically to use the CRISPR-Cas9 technology to create the cancer vaccine and to treat the cancers. In this case, we use the melanoma as one of the example tumor and to test the effectiveness of the vaccines. So this basically cartoon shows what's happening in the tumor developments from the onset to and to pretty much the mature the tumors form and after it starts. So at the very early stages, then when you have a very few tumor cells, then your host immune system try to get rid of those mouse, those tumors, and basically that's in the stage is the immune system win. Then you have some tumors still survive because tumor grows much faster, and then you reach the equilibrium. In this case, the host immune system will not win the fight, and the tumor will start to grow, but not grow too fast, so reach about the equilibrium. And then over the time, those tumors start escape from the host immune system. They start to uncontrolled grow, and that's where the trouble occurred. Now, if we look at the mechanism, what basically is caused escape of the tumors from the host immune system, and the many mechanistic study has been done, the other mechanism has been reviewed, and then we understand there is a several called a do not eat me signal on the surface of the tumors. Particularly, one is the well-characterized do eat not signal as a CD47. So CD47 is the surface markers expressed in all the cells in our bodies, but the level of the CD47 expressions on the tumors extremely high, and that because high level expression of the CD47 protect the mouse and be processed and prevent them to be engulfed by the macrophage. So if the engulf did not happen, that means macrophage would not be able to present the tumor-associated antigen to the T cell, and then the host immune system will not help, will not be able to eliminate the tumors from the body. So what we did, we basically is to try to see what happened if we knock out this CD47. Of course, there is a many study has been done. If you use an antibody against the CD47, you can illuminate the tumor, can shrink the tumor, can extend the lifespan of the cancer patients. And however, because when you use an antibody against the CD47, and because when we're talking about the CD47 expressed in all the cells, including those health cells, and that means antibody also attacked the health health tissue as well. So the idea we have is, can we basically use the CRISPR-Cas9 technology to silence the expression of the CD47 in the tumor cell, not in the health cells? So the idea is basically take the tumor cell from the patient and then grow them in the laboratory, then using CRISPR-Cas9 editing technology to knock out the CD47 expression on the surface of the tumors, then reintroduce this CD47, knock out the tumor cell into the host, and then to see if those CD47 knock out the tumor cell can introduce the protective the antibody or the IgG protections, and that will eventually help the host to fight against the tumor cell. So that's the whole idea. That's the hypothesis. So in order to do these experiments, and we basically constructed, and we take the tumor cell, and in this case, we use the mouse melanoma cells. And what we did, and basically is we used the CRISPR-Cas9 editing technology to knock out the CD47. Here's the results. So this is the unedited melanoma cells, B16F10. We use the Physi conjugated antibody to detect the cell surface display, the CD47. On the melanoma cells, you see this very nicely CD47 expressed, we detected. This is basically is edited. So we did not detect any expression of CD47 on the CD47 on the edited cells. That's indicating the editings occur, and also this the biallelic knockout of the cells. So we also using the flow cytometry to further confirm the CD47 knockout from those cells. And this is basically is negative control and also the isotope control. And this is basically is the negative B16 B16F10. This is unedited cells. So 3BD9, this is the CD47 edited cells. So unedited cell, we detect the CD47 expression. In unedited cell, we did not detect the CD47 expression because this peak overlap with the negative control. So this is basically it's two experiments: fluorescence microscopy for cytometry, very clearly indicating the complete knockout of CD47. Of course, we did a DNA sequencing to further confirm the biallelic knockout. So the next experiment we do, we basically try to confirm whether the So we want to basically is to further confirm whether the macrophage can engulf the CD47 knockout the melanoma cells. So we use the first, we co-culture, we added the CD47 knockout the melanoma cell to the macrophage to co-culture them. And before we added the CD47 knockout the cells, we used the cell tracer to label the cell, in this case, the green. So green labeled is the cell tracer labeled on CD47 knockout melanoma cells, and red color label, that's basically is the macrophage. So when we co-cultured and we see we observed engulf of the CD47 knockout the cells into the macrophage. You see here, there's arrow indicating those are very clear. Those basically, this is the confocal, this is the basically confocal the microscopy imagings. So you see here, there's the red on the indicating the engulf of the CD47 knockout the melanoma cell by the macrophage. So this is the control. We did not observe any engulf of the melanoma cells by the macrophage. So this experiment, this is the confocal microscopy imaging clearly indicating the engulf or phagocytosis of the CD47 negative cells by the macrophage. We also did the flow cytometry, and we did this basically is a negative control, and this is the positive. This is basically is after co-culture the CD47 knockout the cells with the macrophage. So we see in this groups, this is basically indicating, because we detect both the fluorescence emit from the engulfed CD47 melanoma cells and also on the macrophage. And this is basically quantified. So based on this for cytometry data, we quantitatively analyzes the phagocytosis of the CD47 knockout the cells by the macrophage. So the first animal study we did basically using the live and the CD47 knockout cells. We want to see if we transplant those CD47 knockout cells, tumor cells into the mouse, we want to see whether the tumor well developed. Okay, so that's basically to indicating whether those cells can trigger the strong immune response and the host immune system can win the battle and eliminate the tumor cells from and the host. So this is the basic experiment plan. So we implant the both negative control. That's basically is the unedited cells. This is the CD47, 3BD9, that's the edited cells. We implant those cells into the mouse, and we followed almost 60 days to see whether the tumor growth and how rapidly the size of the tumor increase, and whether those the mouse dies. So if you look at it here, for the negative control, and we see the very significantly increase the tumor rapidly, the size of tumor rapidly and grow and rapidly increased over the days, almost about close to about the 18 days, and that's the tumor grow too fat, too large. We have to basically euthanize the mouse. So if you look at the editor, in this case, the vaccine, the 3BD9 inoculated the mouse. We did not see any tumors formed, and we did see one, 2 out of the 10 mouse tested and developed the tumor, but the tumor grow very, very slow, and even 20 days. You see that in the negative growth, most of the mouse and almost all the mouse die, but for the CBD9 vaccine mouse, and the mouse is still alive, and only 2 mouse developed the tumor. And then we also, in those basically mouse did not develop the tumor, we did a second and implant the negative, basically B16F9. We want to see if the immunity generated and in this mouse can protect the mouse from the tumors. So we see half, 50%. So almost 50% of the mouse and can protect the tumor growth, and because of the immunity triggered by the vaccines. Now the next experiment and basically do the tumor challenge. So the first we basically is to using radiation to inactivate the CD47, knock out the 3B9. This is not a live cell. This is basically already inactivated, gamma irradiate, inactivate the cells. And this basically we use the negative control. We do the same thing on the same condition, irradiate both the 3BD9 and the B16F10. And then after 7 days, then we challenge the mouse with the B16F10. This is basically white type melanoma tumor cells. The experiment lasts about 90 days. Then we look at the survival rate, the percentage of the tumor free, that's a survival rate. You see almost about the 70, almost close about 40%, 44% of the 3BD9 vaccinated mouse and keeps the tumor free. And only close about 21% of the B16F10 inoculated mouse well tumor free. So that's basically indicating the CD47 knockout the tumor cells can provide the immunity against the tumors. And that's basically we're right now and doing the further testing and to see and we can increase this 44% to about the 60%, eventually go to the 78%. We basically try to knock out more, and because as I mentioned earlier, the CD47 just one and do not eat me signal. There is several different do eat me signal as well. So we try to knock out them one by one. We want to see if there's a combinatorial effect can more efficiently protect the host from the tumors. So this is basically my collaborator, postdoc, the students work on this project. And this is the funding we received. We received the funding from the NIH and the NSF, NIST, and also some industries. I stop here. We'll be happy to answer your questions.
Okay, great, great, impressive. Let's have some questions then for our speaker. Lorenzo, why don't you go first?
Hi. Well, thank you very much for the talk. It was really amazing. All the data on cancer are really, really nice. So I have a lot of questions, but I will just go on one that I think it will be generic enough. All these things of monotherapy of cancer tend to have problems, I guess, at least in my experience, with the fact that anyway, some cell will evade that monotherapy and just don't mind that particular factor that is involved and just do something else. So to make it short, what would you think about, let's say, those cells that, okay, we convince our immune system to kill all cells that mind 47. And what happens with those cells? They don't mind 47 and they convince macrophages otherwise, and they will survive anyway. And then probably, or possibly give rise to a worse tumor or another tumor somewhere else, etc.
Well, I think it's you brought very important questions. And what is the mechanism? I didn't show you a lot of data because of time constraint, and we did a lot of immunotyping. We look at what happens, how does the tumor microenvironment changes, and how does the spring and response to the vaccines. We did a immunotyping, and we did a T cell priming. We want to understand which T cell is CD47 activated or CD4 activated. We also typing whether B cell also involved in developing these immunities. So because I have not shown you a lot of data, but based on the data to study, actually the paper already published, and I can send you the paper. You can see all the details. So based on immunotyping, we see very clear. So CD8 T cells or T regular cells are basically downregulated. And we also see very significant CD47 and in the host, because after in vaccination, because the host, the tumor cell, still expresses CD47. And then because of the vaccination, the CD47 expression on the host and on host the tumors significantly downregulated and suppress it. And that's probably expand the reason and a macrophage attack those tumor cells, because this vaccine significantly trigger a lot of the tumor microenvironment. So that's basically we studied. So of course, if you look at all this cancer immunotherapy or other treatments, they're all talking about it has to combine with the other, at least the two different approaches in order to reach a high efficiency of the treatment. So we are thinking about, in this experiment, we did not using any adjuvant. We basically just directly injected the mouse. And that's also explain the reason why we only see about a 40%, because this is a very preliminary study. And actually we have a patent pending on this technology. And we, very preliminary study. Right now we are doing more testing and to see if we can bring this efficiency from 44% to 60%, 70%, 80%.
Okay. I was really more on the mechanistic side. I was thinking about, well, I have this fancy about cancer stem cell. I was wondering if these cells might just do something else.
Yeah.
And
well, the vaccine. This vaccine, it do not target to the cancer stem cells. What is a vaccine? What is a vaccine with, you may want to asking, so what is the purpose of this vaccine? People don't, unless, and you basically, the patient develop the tumors. And I think it's the, preventive cancer, vaccine. I don't know how efficient they could be and how effectively they could be. So, however, this technology will be very useful after the surgery. So, for example, if the patient developed the solid tumors, you can use a surgery to remove these solid tumors. That's basically very successful, right? Because very successful treatment for any solid tumors. The question is, because those tumor cell already detach from these solid tumors even before you do the surgery. And those cells, as you say, like very much like a tumor stem cell, they're circulating in the blood, and they're basically staying somewhere in your bodies, waiting for, let's say, after 10 years, those cells were coming back. We call it cancer recurrence. So this vaccine will help against those situations. So basically, when we treat after surgery, if we introduce this CD47 knockout the vaccines, then body will build this immunity. As we say, we already detected the memory T cell. We detect the memory T cell. We detect the B cell mobilizations. That means you have immunities established in the patient. So even if the cancer recurrence occur, this vaccine will help the host to fight against those tumors. So that's basically what I see how this vaccine will be applied in the clinical settings.
Thank you. It was great. Thank you very much.
Yes, thank you. Appreciate it. That's good questions.
Thank you.
Ruchira.
Yes, so thank you for the very intriguing talk and the interesting work. So I had not seen yeast used as a vector for loading a vaccine before. So you had just mentioned with regard to the cancer work about adjuvants. So, and usually, so when one does make a vaccine, then one adds some adjuvant to make the thing more reactive. So is there any possibility that could lead to immunogenicity against just plain yeast?
Yes.
That the person could eventually, become kind of like allergic or something to the unloaded yeast?
Yeah, we did. Actually, we tested this in the St. Jude Children's Research Hospital. They have a WTO influenza virus center. So we tested this vaccine there. So the reason when we use the yeast vaccine, we did not use the adjuvant because the cell wall of the yeast serve as adjuvant. So we tested.
Oh, that makes sense.
Yeah, we didn't see any advantage by adding different adjuvant. We tried a different adjuvant, but we know the yeast alone sufficient to trigger. If you look at HI title, it's reached about 128 titles. So all we need is a 40. So 120 is way above 40 HI title, which we need to completely protect the host from the influenza virus vaccine. So we are currently and work on this technology, try to translate this technology, to use technology to produce the SARS-CoV-2 vaccines. So we are on, and there is an animal study is ongoing. So that's the reason I don't have any data here, and I hope I can present some data. We're still collecting the data right now, and there's animal studies ongoing.
Okay, that helps clear up part of it, but my question was also about like whether the person could develop unwanted.
You're talking about a side effect. So about
yeah.
Right?
Yes.
Yes. The most people do not respond, have their allergy to the yeast, because the yeast that we use is the yeast that's the species we use to brew the beer. So if you can drink the beer and you would not have an allergy to this particularly yeast we used for producing the vaccines. There is some group of the people has allergy to the yeast. Yes, there are, but very small portion of the people populations allergy to the yeast, and that's basically that's why we intentionally choose the yeast, which is used for brewing the beer, and then use this yeast as a host to develop the vaccine. So we're hoping, of course, we did not test the allergy because we did not do the clinical trial, and this basically just animal. So it doesn't help us to determine any allergy, whether there's a allergy to these vaccines.
I see. So if there were a larger trial, then you could determine whether someone would develop the allergy after the vaccine.
That's correct.
I see.
Yes.
Okay.
Yeah, because this is the R01. This project was funded by NIH and through the R01 mechanism. And then when the project ends, and also the influenza virus H5N1s no longer become the issues. So that's why we kind of ran off their funding. We didn't continue to do the clinical trial.
Right, right. There's a whole pipeline.
Right.
Understood. Thank you very much.
Thank you.
Great. Other questions? No? Ruchira, you have another question?
Sorry, I hadn't lowered my hand.
Oh, no, that's fine. Other questions?
I could ask a quick question.
Please, Reed, go ahead.
Let's read. Yeah, both very interesting. I guess I'll just ask about the first question, the first one, and I guess it's about the platform in general. So have you compared it, or maybe not, but what's your thoughts about comparing this platform versus other immunization platforms and its efficacy? So, you know, VLP or other like SAM RNA-based vaccines.
Correct. So when basically the COVID occurred, and we immediately thinking about using this technology to produce the vaccine. So yes, it's compared to the messenger RNA technology, and that may not be so efficient. And if you compare to the adenovirus, right? People talking about the using the adenovirus as a host and to produce the vaccines. But this technology, the yeast, will be compatible, very competitive to the adenovirus as a host, because the reason why this is much better, actually, we are testing. Remember, I mentioned to you there's animal study ongoing. So we are basically testing, because this is the yeast. And with, even using adenovirus, when you transport, how do you basically, after manufacturing, after your production, that you have to ship to the end users. So messenger RNA vaccines require the deep freezer, or at least the -20. So adenovirus-based vaccine require at least a shipping in a cold temperature, at least 4 degrees, not room temperature. But yeast-based vaccine, you can ship them in the room temperatures. So that's
Gotcha.
One is the advantage. The second advantage is the cost. So if you use the adenovirus, then you have to basically use the mammalian cell as a host to produce the adenovirus vaccines. If you use messenger RNA, it's a chemical reactions. But this yeast is just basically yeast. You can use a larger size, larger scale of the bioreactor. You can produce the tons of the yeast vaccines in the low cost, and also the packaging, shipping is very quick. So now regarding the cycles, so the cycles from the virus, from the genome, from recognition of the virus to the vaccine development, that's basically called development cycles. They're pretty much the same as the adenovirus-based vaccines. So as long as we know the sequence, then we will be able to display this protein on the surface of the yeast. Then we can just testing to see if the surface display yeast can be effectively protect the host from the virus infections. So I think it's basically, this is the advantage of using these technologies.
Got it. Thank you.
Other questions? I guess I was going to say, when you talk about cost, in the case of the influenza example, you're using the same vaccine for everyone.
Correct.
In the case of the cancer vaccine, you have to customize it for each patient.
That's correct.
And so I wonder about what can one do to reduce the cost of doing the personalized vaccine development?
Well, the personalized vaccine, because it's a good question for cancer. Yes, we have to, because the genetic variation of the cancer in every host is different, and that's basically the reason. But if you look at the development cycles, and it's also very quick, because as mentioned to you, we can take the tumor cell from the patients, genetically modify, and they take about a month. So it's basically in the second month, we have the vaccine ready. And that's basically the development cycle we're looking at. So I think it's, that's why we said it can handle it. If we want to develop the personalized the vaccine, if it only take about a month, and that shouldn't too bad, and for the any cancer treatment.
I mean, I'll ask one more quick question. Have
Yes,
you considered, just on this topic, because I think it's interesting, and I'm sorry if I missed it. Have you considered like agricultural applications to this, like immunization of livestock and things like that, because then you can avoid some regulatory hurdles?
that's correct. Especially
Yeah.
for the avian influenza virus, that's basically what we're concerned. We try to find a connection.
Yeah. Or foot and mouth disease or something like that. Yeah, yeah.
Yes, you hit this is a very, very good question. We try to find somebody who can help us to do it. Yes.
Yeah,
Absolutely.
Yeah, try different shots on goal. Yeah.
Yes, absolutely. Absolutely. Because you see, this is the yeast, right? They can just mix in the feed in the food which is given to the chicken, can protect them from the avian influenzas. But I don't have a connection. I need I always try to find the connection and reach out to the farmers, agricultures.
Yeah, Luis Rodriguez up at he was at Plum Island, but I don't know if he made it to Manhattan or not. But the USDA folks might be interested in hearing about it.
That would be great. They would be great. We would love to talk to them. So